All right, good morning, ladies and gentlemen. We are going to talk about chromosomal abnormalities today. I'm hoping this one won't take as long as the last couple presentations did. All right, so how can genetic material be changed, and how do changes in genetic material of an organism lead to changes in the traits of that organism? So here are some things that can happen. The first one you're going to need to know about is called nondisjunction. And nondisjunction, I hate the word. For some reason, the word nondisjunction, it's like a meaningless term in my brain, so it's one I just have to memorize. So nondisjunction is something that can happen when chromosomes are aligning at the metaphase plate. So when they're aligning in the middle and they're getting ready to be split. So nondisjunction can take place either, look at the first example on the left side, when you have two homologous partners and they need to go to opposite sides of the cell. When nondisjunction happens, instead of going to opposite sides of the cell, one set of the homologous partners both go to the same side of the cell. And so that leads to, look at, if they're skipping a whole bunch of steps here, but if you look at the sex cells at the bottom, they end up with the ones where the homologous chromosomes stayed stuck together, they end up with an extra chromosome in each of those sex cells. And then in the other sex cells, because they lost, see, they were supposed to be a chromosome right here, they end up missing a chromosome. So now if that, let's say those are for sperm. If one of those, if the sperm that has the extra chromosome then fertilizes an egg, let's use humans as an example. So this would have 24 chromosomes in it, and it fertilizes an egg that has 23 chromosomes in it. You end up with an organism, instead of having 46, or a human, instead of having 46 chromosomes, it has 47 chromosomes. That is called a trisomy. What that means is that individual has 3 copies of the same chromosome. And when that happens in a sex cell, that leads to an individual, the offspring then, always has some kind of a syndrome associated with it. Down syndrome is like the most famous one, the one that most people have heard of. But many of the trisomies are deadly. So with humans and with many vertebrate species, we need exactly the right number of chromosomes. And if anything screws that up, it can lead to all kinds of genetic disorders or even the inviability of the fetus itself, so miscarriage. The other time, well, there's 2 other times that nondisjunction can take place. Take a look at the other example on the right side. This time, the homologous partners separated correctly, but then notice that the bivalent chromosomes, they were supposed to separate to opposite sides, and one of the bivalent chromosomes didn't separate. So notice that this cell line, these guys divided correctly, these guys divided correctly. So we'll say these are sperm. The sperm have the right number of chromosomes in them. And let's make sure you're clear on that too. The original cell, which is called a germ cell, and again, that's just one of those words you kind of have to memorize. I usually think about the letter G. Germ cell is what germ cells divide to form gametes, which also has a letter G in it. So germ cells divide to form gametes. So the original germ cell had 4 chromosomes. They happen to be bivalent because they were copied, but it is 4 chromosomes. If you look down at the sperm, they correctly have half the DNA. So this is the diploid number. This is the haploid number. So they have half the DNA, and that side of the cell line went great. But on this side, the homologous partners separated correctly, but then one of the bivalent chromosomes didn't pull apart, and it stayed and went to one side, and that led to one of the sperm cells being having an extra chromosome, and one of the sperm cells having a missing chromosome. All right. And the other time that can happen, it could happen in a non-fetus. So it could happen, not in a sperm or egg cell. I didn't say that well. It could happen later in development once the cells are just going through mitosis. It can happen right here also. But in that circumstance, you have far less impact on the total organism. So it's not as important. If this happens, if if the sperm or the egg have the wrong number of chromosomes, it leads to a fertilized egg, which is called a zygote. Ladies and gentlemen, the fertilized egg is called a zygote. It has the wrong number of chromosomes, and then every cell that forms from mitosis from that zygote is going to have the wrong number of chromosomes. And so that means this person that forms is going to have a syndrome in every cell of their body. If it were to happen in KJ's body right now, I'm already made of 100 trillion cells that don't have that disorder. So if one of my cells goes awry and none of my checkpoints fix it, it might divide and give me, I don't know, 50 cells that have the wrong number of chromosomes. It'll never be that big of a number anyway. But if I have the wrong number of chromosomes in just a tiny number of my cells, that has no impact on my body. So the time that it's really important is when first, if it happens in a sperm or an egg, and then if the embryo is really, really, really early on in development, so 4 cells, 8 cells, 16 cells, that's when one of these non-disjunctions can really be a problem. So this leads to gametes with irregular number of chromosomes. And then it says this must happen in a germline cell to have an effect. And he's just talking about, the author of this is just saying the germ cells are the ones that are going to produce a full body syndrome, as opposed to me getting a few wrong chromosomes in a few of my cells. It's not going to be a syndrome for me. Okay, moving on. There are all kinds of giant mutations that can take place within a chromosome. There are also tiny mutations. We're going to talk about the giant mutations first, but let me just, not first, second. Let's just change the subject for a second. We're going to learn in our next unit about DNA and the letters of DNA, and most of you know the A, T, C, and G. It is possible to just change one letter, one A, T, C, or G, and that would change the amino acid that is supposed to be put there, which changes the conformation of the protein, which could lead to a genetic disorder. That's called a point mutation. It's just a teeny tiny little mutation. Most of the time we have point mutations and it's no big deal. Sometimes we have point mutations and it leads to genetic disorders like sickle cell anemia, cystic fibrosis, it could even lead to cancer, depending where that point mutation takes place. These mutations are giant mutations. This is where thousands, I'm making up a number, thousands of A's, T's, C's, and G's get rearranged, deleted, copied. And so these are called large-scale rearrangements. So segments of chromosomes can also get altered during DNA replication. Big screw-ups can happen. So there are 4 major types, and fortunately, they have some pretty good names that make it pretty obvious. The first thing, so this is a chromosome right here. Notice an entire section of the chromosome is missing. I believe there's a genetic disorder called cri du chat, which means cry of the cat in French. And it's caused by the deletion of a deletion of part of one of those chromosomes. So that's one example. Duplication, you can actually copy parts of chromosomes accidentally. And we saw that with the proto-oncogene. Proto-oncogenes are supposed to tell the cell when to divide. But if we have a mutation that leads to lots of proto-oncogenes that aren't supposed to be there, the cell is going to keep dividing because they're going to keep telling the cell to divide. So that would be an example of a duplication. Easy word, it makes sense. To invert something means to flip it over. So look at the 2 examples, the one above and the one below. Notice that B, C, D are now in the backwards order. What's interesting about this particular mutation, that's not the right word, this chromosomal damage, whatever, rearrangement, is that they're technically still intact. The genes are still intact. So it's possible that the person will have no impact from this whatsoever, that they will have no, they call it a silent impact, that the person won't even know that they have an inverted gene. And then the last one is called translocation. And translocation is translocation trans means to move, and locations. So a part of a chromosome breaks off, no kidding, this can really happen, and becomes reattached to a different chromosome. Now, this is a double translocation, which I've never seen before. I'm surprised that they drew this as a diagram. Usually what you see is just one chromosome that's missing a section and one chromosome that has an additional section added onto it. But that's called translocation in that circumstance. Give me a second here. Whoops. Okay, already did that. So let's talk about some examples. So anytime that, let's just go with humans for right now. Anytime that humans have either the addition or the subtraction of a single chromosome, it's either called a trisomy, that means they have 3 of one chromosome, or it could be called a monosomy if they're missing a chromosome. So then they only have one of their pairs of chromosomes. It's usually results in a miscarriage. So almost always these the somies are deadly or have massive impacts on the organism. Trisomy 21 is one of the survivable trisomies. Part of the reason, well, I'll let you look at it. It says, down at the bottom, it says, why would an extra 21 be tolerated by the organism? Take a look at the karyotype and look what is notable about chromosome 21, and if you had a third chromosome 21? Giving you a moment to think. Yeah, it's really, really, really small. So having a small chromosome 21 tends to be less dangerous or less harmful than having, say, a third chromosome 1, which is our largest chromosome. All right, moving on. Oops, every time. Okay, it is possible to have some somies that have to do with the sex chromosomes, and that's always, students usually find these super interesting. Turner syndrome is monosomy X, which just means that they have one X chromosome. For the most part, they don't have a huge impact. They might have infertility issues, sometimes some heart defects, but otherwise you wouldn't know if you wouldn't know if a woman was monosomy. They wouldn't know that they were monosomy X because females have 2 X's, and one of the X's always inactivates, which we're going to talk about in just a second. This is a very survivable condition. Klinefelter's is a super interesting condition. This is where males who typically biological male is XY, but for any number of reasons that occur, this is nondisjunction, where the chromosomes are staying stuck together, it leads to extra X chromosomes. Ironically, in the karyotype that they show here, this person has 4 X chromosomes. Usually we think of Klinefelter's as having 2 X chromosomes. So what's interesting about individuals with Klinefelter's, they are boy in every way, shape, and form. They have the Y chromosome. They look like boys. They have boy genitalia. However, the X chromosome, the extra X chromosome, leads to what are called secondary sex characteristics that are female. So these boys, these poor boys, end up developing breast tissue, their voices don't lower, their beards don't grow. And so it can be a very, very, it's not at all associated with feeling like you're in the wrong body. That's a different issue. It's just associated with having kind of mixed sex traits, sex characteristics, but not necessarily with anything else about gender or anything like that. All right, next, multiple Y chromosomes. So having an extra Y chromosome doesn't seem to be a problem in the same way that trisomy 21 isn't a terrible problem. And that's because the Y chromosome is wee. It is a teeny tiny little chromosome. And so it just doesn't have too much impact on any individual that has an extra Y chromosome. And then finally, multiple X. The X chromosome is a very, 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 very large chromosome. It contains lots of genes. But both males and females have to function with it, and males only get 1 copy of it. So how is it that people survive with multiple X chromosomes? Well, it turns out there's this process, and we're not going to go into a lot of detail, but it is super interesting, that's called X inactivation. Early in development, every X chromosome in a cell, except for one, is turned into an inactive, and they call it a bar body. So take a look at this because it's so interesting. This calico cat example, let me bring it up closer here. So this calico cats have X chromosomes, just like X and Y chromosomes, just like humans do. And so this is a female cat because it received 2 X chromosomes. One of the X chromosomes, so it's random in all of our cells. It's random which of the X chromosomes, so these are in females, I guess, anybody that has more than one X chromosome, it's random which ones become bar bodies and which ones become inactivated. So this cell right here might have my dad's X chromosome inactivated, and this cell right here might have my mom's X chromosome inactivated. It varies all over the body. So for a calico cat, this particular calico cat has an allele for orange fur and an allele for black fur. In some of its cells, the orange was inactivated. In some of the cells, the black was inactivated. And so you end up with a cat that is a patchwork of black and orange. And they are always female cats. You can just tell by looking at them that they are female cats. So here's something I hope is a fascinating puzzle to you. I have just gotten done teaching you that females are less likely to develop sex-linked traits, sex-linked disorders than males are. And so the question is why? And it is utterly fascinating, the answer to this. So if our X chromosomes inactivate, then doesn't that get rid of the female advantage? And that is an area of super cool study right now. So what I teach you is that you have this backup copy of the X chromosome, which is true. However, they get inactivated. 50% of the time, the X chromosome that's being inactivated might be your backup copy, might be the good copy, the copy that doesn't have red-green color blindness. So why don't more women show red-green color blindness if their X's are being inactivated? Well, it turns out, and I could give you an article on it because it's super interesting. It turns out that having 50% of the ability to see color is good enough to see color. So these women who are carriers of the red-green color blindness, half of their normal X chromosomes, their full vision chromosomes, are working. Half of their colorblind X chromosomes are working. And so they're technically not full color vision, but it's good enough, and they're able to distinguish between red and green color. And there's all kinds of other things that are super interesting about that we could talk about some other time. All right. Ploidy. So the ending ploidy refers to how many chromosomes are in the entire set. So haploid cells, as we know, in like humans, for example, haploid cells are the sex cells. Diploid cells are all the body cells. It is possible to have something called triploidy, which would be 3 sets of chromosomes, or even tetraploidy, and even beyond that. So they refer to triploidy, tetraploidy, and beyond as polyploidy. And you right now have a learning target that is specifically about the advantages or disadvantages to being polyploid. So in most vertebrates, we cannot be polyploid. We have to be diploid, and having the wrong number of chromosomes leads to a terrible syndrome. But it turns out that there are many species that are polyploid, and that is an actual advantage to them. And I want you to think about why that might be an advantage. Think about the advantage that females have having that backup X chromosome. So it's a similar idea here. When a species is triploid or polyploid, if they have a chromosome that's not functioning for whatever reason, they have other chromosomes that they can rely on, other copies of that chromosome that they can rely on, that make them survive better or have better traits. Yeah. Okay, so moving on. Oh, I just started something. I didn't mean to do that. Okay, rearrangements. There is no rule about how a chromosomal rearrangement will affect an organism. Sometimes it's detrimental, sometimes it's silent. Same thing with those point mutations. We could change one letter of our DNA and have no impact whatsoever. We could change one letter of our DNA and develop cystic fibrosis or sickle cell anemia. So we call that silent when nothing happens. And that's it, you guys. There's nothing else for us to talk about. These are just some examples, but blah, blah, blah.